And we are live. It's 12:05 p.m. Eastern Standard Time on Friday, August 18th, year 2023. This is Houstonic.live. My name is Mark Kulak, and today we have a part two of an audiobook that we are playing from Amazon, Audible, if you will. Gene Machine, 2019 book by Venki Ramakrishnan. Yesterday, we listened to the intro, prologue, if you will, all the way up to the end of Chapter 5, titled Going to the Mecca of Crystallography. And today, we will be continuing the book. Excuse me here. I have to just get a couple of things set up here. I don't know why. It's one little thing I wish that they would change on the video streaming software that I use. And that is the ability to turn the camera around, to use the face camera before I have to make it live. But whatever the case, today we are going to be listening to Chapter 6 and onward to as much as we can. There's still a total of about 6 hours of audio left in this book. It's a fantastic book so far. We are listening to this because the Houstonic Live audience is educated, advanced, curious, and we collectively are coming to the realization that there's much more to the human genome than just DNA. Sort of like saying there's more to computers than just the binary code. And very few people, almost no other channels actually, sadly, are talking about the ribosome, are talking about the processor that converts the code to things, much less also how proteins themselves function. Sort of like a dual stage, if you will, translation and discussion of individual genes, while certainly toying around with those settings, if you will, those states in certain situations is going to have a ripple effect, undeniably. It's also not debatable that until you understand the whole chain of events, who really knows what causes what or how and what the ripple effects really are. And so this is part of one, getting us to collectively understand that statements that are being made about the knowledge of how to flip individual genes around can cause or cure problems. And to disarm some of those arguments and to help people deal with the fear that is running rampant because of such statements. But more so to have a more broad understanding and in particular, one of this particular topic of the ribosome, which there was a quick introduction given to so far in this book by Venki Ramakrishnan. And also, last but not least, there is a hypothesis and that we are standing behind here on Houstonic.live, that much of what's happening right now is I shouldn't just say an attempt to collect data. That's sort of like saying if you get everyone's DNA, you're all set. It is to create a society where as much data can be collected in as many situations as possible, about as many people as possible. Daily DNA, hourly DNA, from as many different cell types as possible, because it's not like you just have one DNA that all the cells reference. Also, to be able to monitor vaccines, to be able to monitor health, to be able to standardize the food supply. You just don't have your lab rats and give some food from one place and the other one's food from another place. You need to standardize it. You need to standardize your assets. As Greg Simon, the one of the founding directors and previous executive director of the Biden Cancer Moonshot Initiative, said, it is the patients that are the assets. In particular, it's the ability to collect data from the patients that are the assets. So with that said, that lays the groundwork for why we are listening to this book. I think Venki is, so far, from everything I've seen, I think he's a solid guy. He got married relatively young. He adopted the 4-year-old girl that was the daughter of the woman that he married, and has had one other child since, which may not seem too pertinent at first, but to me, I want a guy that is going to make commitments and is in it for the long run, regardless of how hard it is. Little things like that appeal to me, okay? Maybe not everyone, but for me, little things like that. Sure, he has a bit of a belief of that's not necessarily great of the United States and its ability to willingness to invest into sciences and so on. He's got some points there, but that said, it's not a matter of being in political harmony with Venki Ramakrishnan. I just think that his willingness to or desire to share his story, what he went through, is also helps us better understand this gene machine, the ribosome itself, but also the history that led us up to this. I don't see so far an agenda to this book, okay? I don't see an agenda. So far, I'm hearing the voice of someone who I think would be pretty cool to get to know. It won't happen, but I think it would be pretty cool. He's also the first non-English born person to be the president, if you will, or the head of the Royal Society. That's a big deal. A 300-year-old society, and he was the first non-native born person. So it's kind of a big deal. Anyways, with that said, we're just going to click play. As I've told my owners before, my life has changed a lot lately. I have 3 to 4 hours a day right now of additional responsibilities that go along because of life circumstance changes. It's tough. We make do. I share this not because my story is unique. My story is actually not unique. That's why I share it. Because we are, I want everyone to see what is happening to millions of people within our country, the United States right now. Yes, I'm U.S.-centric. Doesn't mean that I'm U.S. elitist, though. Hopefully you feel, if you're not in the United States, as strongly about your community as I do about mine. I don't really have much room left in my heart to feel anger and fear towards other countries. Right now, I'm just trying to get my own straightened out. So with that said, everyone, please enjoy. This will probably be playing for a couple of hours, and I won't be here, but I will be playing this probably again, sitting in the chair, starting and stopping, pausing, doing some researches along the way. This is just my attempt to be able to capture it all, share it with you, and etc. So with that, no further ado, no further blah, 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 blah. Some people say I ramble a little bit too much. Here we go, on to Chapter 6 of Gene Machine 2019 by Venki Ramakrishnan.
Chapter 6. Emerging from the Primordial Mist. During my sabbatical, 2 papers made me rethink my approach to the ribosome. One of them dealt with a long-standing chicken or egg paradox about how the ribosome could even have emerged. All life today depends on the thousands of reactions carried out by proteins. The ribosome, the machine that makes proteins, itself consists of lots of proteins. So, how could the ribosome have even come into existence? Once again, it was Crick who had a key insight. In a classic paper in 1968, he was struck by the fact that although ribosomes had lots of proteins, they were made up mainly of RNA. What was the purpose of this ribosomal RNA? Crick suggested that ribosomal RNA and tRNA were part of the primitive machinery of protein synthesis, emphasis in the original. He then went on to say, it is tempting to wonder if the primitive ribosome could have been made entirely of RNA. The problem was that when Crick proposed this, all known enzymes, the biological molecules in every cell that could carry out the chemical reactions that are essential for life, were proteins. Nucleic acids like DNA or RNA could at best be inert carriers of information. There was not a shred of evidence that RNA could carry out any kind of chemical reaction, let alone direct a complicated process like translating genes into proteins. By then, other enzymes involving DNA and RNA had been discovered. ones that made copies of DNA during cell division or others that copied DNA into messenger RNA. They were all made up of proteins. So the general feeling was that ribosomal RNA was just a kind of scaffold on which to hang the various proteins, each of which would do one of the many jobs of the ribosome. One protein might help tRNA to recognize the code, another could help add amino acids to the growing protein chain, and so on. That explained why there were so many ribosomal proteins. Early work on antibiotics supported the idea that it was the proteins that were performing the many important tasks in the ribosome. It had been known ever since the 1950s that many antibiotics worked by blocking the ribosome. Nomura showed that mutant bacteria that were resistant to streptomycin had an altered ribosomal protein. Mutations in other proteins also changed how the ribosome responded to antibiotics. If changes in proteins were making the ribosome behave differently, it must be because they were doing something important. Although lots of scientists were focusing on the proteins, a few were intrigued by ribosomal RNA. One of them was Harry Noller, whom I would jokingly refer to as the Sage of Santa Cruz. With his long hair and beard, typically wearing jeans with a t-shirt, he superficially had the demeanor of a mellow, pot-smoking California hippie. He also had a fondness for motorcycles and antique Ferraris. Even his computers were named after Italian racing car drivers. His cool charisma and wry wit meant that in public gatherings, he was sometimes surrounded by adoring young scientists, much like groupies around a rock star. He had a large cult-like following among his many protégés, who viewed him as the spiritual leader of the ribosome. But underneath it all, he was a serious and deeply ambitious man with a relentless focus on the ribosome. A California native, Harry got his undergraduate degree at Berkeley before going on to get his doctorate in protein chemistry from Oregon. Then he too became a postdoctoral fellow at the LMB. There, under Ewan Harris, he was working on a protein that is involved in metabolizing glucose. In an autobiographical essay, he describes how he was feeling slightly intimidated by the whole scene at a party in one of the Cambridge colleges, when Sidney Brenner came up to him and asked him who he was and what he was doing. On hearing that Harry was working on glyceraldehyde phosphate dehydrogenase, Brenner announced, "That's stupid. If you're a protein chemist, why don't you work on something interesting, like ribosomes?" In hindsight, this is quite ironic, considering Brenner thought ribosomes were not interesting enough to pursue at the LMB himself. Harry was initially crushed by this scathing assessment, but then, in a bold decision, he decided Brenner was right and left Cambridge to work with Alfred Tessier in Geneva. There he overlapped slightly with Peter Moore, who was doing the same European tour in the opposite direction, going to Geneva first and then to Cambridge. Peter said that Harry was hired in Geneva for his protein expertise, so they could purify and characterize all the ribosomal proteins. After all, that must be where the action was. When Harry went back to California and started his own lab in Santa Cruz, he did a key experiment that changed the course of his life. He and his student, Jonathan Chairs, showed that if you modified the ribosomal RNA in the small subunit with a chemical called cathoxyl, it could no longer bind tRNA. This was the first clue that ribosomal RNA actually might have an important function. Many scientists might have dismissed it as a curiosity, but to Harry's credit, he followed this up and became a lifelong RNA biologist. Then, in the early 1980s, there was a scientific earthquake initiated by two scientists, Tom Cech in Colorado and Sidney Altman at Yale. Cech was looking for the enzyme that carried out a reaction in which a stretch of RNA could excise itself from a longer piece. He found that just the RNA could excise itself without any help from a protein. Altman, on the other hand, was studying the properties of an enzyme that could cleave certain RNA molecules. The enzyme itself was a complex of a protein and RNA, and to his surprise, he found the RNA component by itself could carry out the cleavage reaction. So both groups had shown that RNA by itself could carry out a chemical reaction. Enzymes that were made of RNA came to be called ribozymes to distinguish them from the more usual protein enzymes. Although the reactions seemed specialized, they have huge implications for the origin of life. How life began is one of the great remaining mysteries of biology. All life requires some form of energy in the right chemical environment. Some people have pointed out that a lot of the chemistry that life uses resembles the kind of chemistry that occurs at the edges of geothermal vents in the ocean. Even if this is merely a coincidence, as others have argued, it is useful to think about what conditions made it possible for life to emerge. But fundamentally, life is more than a set of chemical reactions. It is the ability to store and reproduce genetic information in a way that allows complex life forms to evolve from very primitive ones. By this criterion, there is no question that even viruses are alive, even though people used to question it because they need a host cell to reproduce. However, anybody who has become ill from a virus and experienced his or her body fighting an infection would not doubt that viruses are alive. The problem was that in nearly all forms of life, DNA carried genetic information, but DNA itself was inert and made by a large number of protein enzymes, which required not only RNA, but also the ribosome to make those enzymes. Moreover, the sugar in DNA, deoxyribose, was made from ribose by a large complicated protein. Nobody could understand how the whole system could have started. Scientists who were thinking about how life began, like Crick, Leslie Orgel at the Salk Institute in La Jolla, and Carl Woese at the University of Illinois, suggested that maybe life began with RNA. At the time, this was pure speculation, almost science fiction, because RNA was not known to be capable of carrying out chemical reactions. Czech's and Altman's discovery changed all that. RNA was now a molecule that could both carry information as a sequence of bases, just like DNA, and could also carry out chemical reactions, like proteins. We now know that the building blocks of RNA can be made from simple chemicals that could have been around in the Earth billions of years ago. So it is possible to imagine how life may have started with lots of randomly made RNA molecules until some of them could reproduce just themselves. Once this happened, evolution and natural selection could allow more and more complicated molecules to be made. Eventually, even something as complicated as a primordial ribosome. The idea of a primordial RNA world, a term first coined by Wally Gilbert, became widely accepted. The ribosome may have started off in an RNA-dominated world, but because it made proteins, it became a Trojan horse. Proteins turned out to be much better at carrying out most kinds of reactions than RNA because their amino acids are capable of more varied chemistry than the simpler RNA molecule. This meant that as proteins were made, they gradually evolved to take over most of the functions of the RNA molecules around at the time and much more. In doing so, they transformed life as we know it. This may also explain why, although the ribosome has a lot of RNA, the enzymes that replicate DNA or copy it into RNA are now made entirely of proteins. This is probably because the use of DNA to store genes came later. By that time, proteins had become prevalent and were carrying out most of the reactions in the cell. Of course, this doesn't explain how genes carrying a code to make proteins came into being. The best guess is that an early form of ribosomes just made short stretches of random peptides, which helped to improve the RNA enzymes that were around then. But from there, how genes originated that carried instructions to make proteins that had amino acids strung together in a very specific order was quite a leap, and is still one of the great mysteries of life. And that, in turn, would mean that in addition to the large subunit, many other elements would have to come into existence. mRNA to carry the genetic code, tRNAs to bring in amino acids, and the small subunit to provide a platform for the mRNA and tRNAs to bind. But before the discovery of RNA catalysis, people couldn't see how the system could have begun even in principle. Why could RNA carry out reactions and not DNA? The main difference between the two molecules is just an oxygen atom on the ribose sugar of RNA to form a hydroxyl OH group. We now know that this small difference allows the OH groups from different parts of the RNA molecule to bond with each other, so RNA can fold back on itself and have compact, three-dimensional shapes like protein enzymes, with pockets to carry out chemical reactions. After Czech's and Altman's discovery, everyone realized that Crick was probably right in suggesting that a primordial ribosome was entirely made of RNA. What about today's ribosome? Had key functions been taken over by proteins as they had with other enzymes? Or, as now seemed completely plausible, did ribosomal RNA still carry out most of its important functions. Meanwhile, Harry had continued his work on ribosomal RNA. He had no idea of the location of the chemical modifications that prevented tRNA from binding the ribosome. In fact, at the time, nobody even knew the sequence of ribosomal RNA. Soon after Harry's initial results, Fred Sanger at the LMB had figured out how to sequence or determine the precise order of the bases in any given piece of DNA, for which he received his second Nobel Prize. He is one of very few people to have won two. So Harry returned briefly to Cambridge, this time to learn how to sequence DNA. Instead of trying to sequence the RNA directly, which remains a much more difficult task, Harry used Sanger's methods to determine the exact sequence of ribosomal RNA by sequencing their genes, which resided on DNA. The large ribosomal RNA from the 30S and 50S subunits are called 16S and 23S RNA, respectively. Sequencing ribosomal RNAs turned out to be important by itself. By comparing sequences from different species, Carl Woese and Harry could work out how they were related and how the RNA molecule would fold back on itself so that parts of the molecule formed internal base pairs with other parts. This internal base pairing meant that ribosomal RNA had lots of segments that were double helical. Eventually, comparing ribosomal RNA sequences led Woese to discover that in addition to bacteria and eukaryotes, there was a third distinct domain of life called the archaea. It is now believed that an early bacterium joined an ancient archaeon to form the first eukaryote, whose cells have a nucleus. Archaea, like bacteria, are prokaryotes, meaning they have no cell nucleus. Eukaryotes then evolved into the complex multicellular organisms we have today, including humans. Once the sequence of ribosomal RNA was known, Harry could then try to locate where on the RNA molecule his chemical agents modified it. He adapted a technique that people had developed to see where proteins would bind on DNA. Scientists would expose DNA with a chemical that modified it, then do the same when a protein was bound to it. The protein would protect those parts of the DNA that it bound because the chemical couldn't reach them. Afterwards, they could compare the modifications in the DNA with and without the protein, and the difference would tell them roughly where the protein sat on the DNA molecule, or its footprint. Harry and his students, most notably Dinesh Moazad, started applying this method, called footprinting, to ribosomal RNA. They figured out which parts of ribosomal RNA were involved in binding to tRNA molecules and to each of the ribosomal proteins. This technique produced a lot of data on what touches what in the ribosome, but like other studies carried out in virtually every other lab in the world working on the ribosome, it did not produce a real sense of how it all fitted together, let alone how it functioned. Doing footprinting with antibiotics was more interesting. Many antibiotics bind to ribosomes, but even though some altered proteins made ribosomes resistant to antibiotics, nobody could get antibiotics to bind to any of the ribosomal proteins by themselves. By footprinting, Harry was able to show that each antibiotic bound to a particular part of ribosomal RNA. Since antibiotics stop ribosomes from working, clearly ribosomal RNA must be doing something important. Because of Czech's and Ortmann's discovery that RNA could carry out reactions, and Harry's work on antibiotics, the field was quickly coming around to the view that ribosomal RNA played an important and possibly central role in the ribosome. In any case, after being passé for a long time, ribosomes became interesting again. In a prophetic article in Nature called The Ribosome Returns, Peter Moore wrote in 1988, "Fashions come and go in biochemistry. The discovery that some RNAs are enzymes is reviving interest in the long-neglected ribosome." Not even he could have predicted it would return with such a vengeance. Towards the end of my sabbatical in 1992, a paper in Science by Harry generated a lot of excitement. He tried to settle the question of whether just the RNA part of the ribosome could carry out a key reaction of the ribosome called peptidal transfer, or the joining together of two amino acids to form a peptide bond. In other words, was the ribosome a ribozyme? He took 50S subunits from a species of thermos bacteria that grow in the hot springs of Yellowstone National Park and treated them with an enzyme that digests and degrades the proteins into fragments. He then extracted as much of the remaining protein fragments as he could. The resulting subunits consisted almost entirely of RNA and could still carry out the reaction. Harry's paper generated quite a buzz among scientists broadly, but by then the result was not a huge surprise to the ribosome community. It was also not definitive. Harry had gone to great trouble to chew away and remove the proteins in the 50S subunit, but there were still lots of protein fragments and even some whole proteins left in his particles. So, the reaction could still have been carried out by a protein or a piece of one. When Harry used a different method to remove all the proteins completely, the particles were no longer active. The procedure didn't work with ribosomes from E. coli, which is what most people studied. Harry himself implicitly acknowledged the limitations of his work by his rather cautious title, Unusual Resistance of Peptidyl Transferase to Protein Extraction Procedures. A few years later, in 1998, a group in Japan thought they had nailed it when they made pure fragments of ribosomal RNA that could carry out the reaction. But after publishing this with much fanfare, also in Science, they found flaws in their work and withdrew their paper a year later. It was clear that after 40 years of trying to solve how the ribosome worked by chemical methods alone, other means would be needed. In the same paper where Crick suggested that an early ribosome might have been made up entirely of RNA, he also said, "Without a more detailed knowledge of the structure of present-day ribosomes, it is difficult to make an informed guess." Chapter 7. A Threshold is Crossed. Apart from Harry Noller's paper in Science, the other paper that hit me during my sabbatical was a short report in 1991 by Ada Jonath on a dramatic improvement with one of her crystals of the large subunit. For the first time, at least in principle, the crystals were good enough to obtain an atomic structure of an entire subunit of the ribosome, with hundreds of thousands of atoms. A threshold had been crossed. To understand when a crystal is good enough requires some explanation. So far, we've assumed that crystals are formed by molecules packing identically into a three-dimensional stack, also called a lattice. This rarely happens with large molecules like proteins. During crystallization, a protein molecule finds its way into the growing crystal lattice, but because it is large and floppy, it doesn't sit in exactly the same orientation as its neighboring molecules. The final image is the result of adding up the contributions of the millions of individual molecules in the crystal. If the molecules are all sitting slightly differently in the crystal, their contributions will be blurred out to a greater or larger extent. Think of what a multiple exposure photo of something immovable, like a rock, would be, compared to a multiple exposure of a person who cannot sit perfectly still. How good a crystal is depends not on how nice it looks visually, but how well it diffracts X-rays. As I pointed out in Chapter 3, resolution tells us how close two features can be and still be distinguished. In practice, you can tell how good the resolution of a crystal is by seeing how far out in angle the X-ray spots extend from the direction of the incident beam. In the worst crystals, there are only a few diffraction spots around the main X-ray beam. If you solved the structure from that sort of crystal, you'd only see a blobby-looking object with almost no detail, but it might give you a sense of its overall shape. For more moderate crystals, which yield data to, say, 5 to 7 angstrom resolution, a unit of angstrom is 0.1 billionth of a meter. You could see some characteristic features of proteins and DNA or RNA. For example, you would just be able to see the grooves in the helices of DNA or RNA. The narrower single-chain alpha helices adopted by many proteins would look like tubes. In a well-diffracting crystal, the spots can be seen out to very high angles, all the way out to the theoretical limit imposed by the wavelength of the X-rays. Remember, you cannot see features that are closer than about half the wavelength. At that limit, when you obtain maps by solving the structure, you can see individual atoms as distinct spheres. This is often seen for simple molecules like salts, which have only a few atoms and have precisely the same orientation in a crystal. But even small proteins almost never reach 1 angstrom resolution, like small molecules, and as the molecules get larger and floppier, it is very rare for them to produce crystals that diffract to very high resolution. In practice, this means the diffraction spots get weaker and weaker at higher angles, and then just disappear. So the information about fine detail is lost because there isn't enough order in the crystal to produce it. The higher the angle to which you can see diffraction spots, the higher the resolution of the structure you can get from the data. So crystallographers typically talk about the resolution of the crystal or even of a particular diffraction spot. As the resolution improves beyond 3.5 angstrom, you are still nowhere near being able to see individual atoms, for which you need to be better than 1 angstrom. But you can still obtain an atomic structure because you can start to see the characteristic shapes of amino acids and bases. In proteins, amino acids vary in shape from large and flat to long and thin to short and stubby. If you know the order in which they should appear, then you can fit a protein chain with these amino acids into their shapes, much like solving a large three-dimensional jigsaw puzzle. Similarly, for DNA or RNA, the bases T, or U, and C are small, and the bases A and G are large. So even though you can't see the individual atoms, you can build a chemical structure in atomic detail in these maps. If there are amino acids of very similar shapes near each other in the sequence, it is still possible to make mistakes because, unlike a jigsaw puzzle you buy in a store, the shapes in a three-dimensional map aren't perfect due to errors in the data. Also, unlike a jigsaw puzzle, this problem is in 3 rather than 2 dimensions, and there is no answer conveniently provided on the front of the box. So, the threshold of about 3.5 angstrom is the holy grail for people trying to get crystals. Anything better means you have a good chance of solving an atomic structure, whereas worse than about 4 angstrom means that it won't be easy unless you already know roughly what the molecule looks like. The original crystals of ribosomes were terrible. They produced hardly any diffraction spots at all. Rather than being discouraged, Ada Yonath, now working with her long-term associate, Francois Franceschi, who was overseeing the biochemical operations for crystallization in the Berlin Institute, continued to toil away in a systematic effort to produce better crystals, even though there was no clear prospect of a structure from them. Ada was also carrying out some of this work in her lab at the Weizmann Institute in Israel. At the time, many scientists in Israel were identifying new species to characterize the local biodiversity, and had identified a microorganism called Haloarcula marismortui, which, as its name marismortui suggests, literally sea dead, grows in the extremely salty Dead Sea. Because it grew in a very high salt environment, rather than very high temperature, it represented a different kind of extremophile, organisms that grow in extreme conditions. Later, it turned out that Haloarcula is not a bacterium, but an archaeon. Its ribosomes are more complex and in some ways halfway between those of a bacterium and a eukaryote. Ada thought its ribosomes might be worth trying to crystallize, and the large subunits from it yielded better quality crystals than any of the others. Initially, they were not good enough to produce an atomic structure, but by tinkering with the conditions in which the crystals grew, Ada's group was able to improve them. Apart from getting good crystals, there was a major problem with actually collecting data from them. Crystals of many proteins, and especially of large molecules like the ribosome, would be damaged by the X-rays themselves before the data could even be collected. These data are typically collected by rotating a crystal in a beam of X-rays and taking a series of snapshots to measure the scattered X-rays with a detector. For each orientation, some of the planes of the crystal will satisfy Bragg's law and produce spots in certain directions. When you collect all the possible spots this way, you have a complete data set and can start calculating your structure. Ribosome crystals were particularly difficult to work with because the diffraction spots from them were so weak. This is because the larger the molecule, the fewer of them there are in a crystal of any given size. Since the strength of the spots is related both to the number of molecules, because they come from adding up the contribution of the individual molecules, as well as the intrinsic quality of the crystals, the diffraction spots from ribosome crystals faced a double handicap and were much weaker than those of typical protein crystals, let alone salt crystals. To see them at all, they have to be exposed to a very intense beam of X-rays for a long time. During this time, the X-rays damage the molecules and create disorder in the crystals. As a result, the molecules break up partially, and either their internal structure changes or they no longer sit precisely in the same orientation as they did originally. To make matters worse, the initial damage generates free radicals that spread through the crystal and cause even more damage. With a large crystal, you can often visually see where the X-rays have damaged the crystal because the free radicals generated by X-ray damage often have a faint but distinct coloration. The result is that the resolution of the crystal falls off with time during the experiment. The loss of resolution is directly visible because the diffraction spots at high angles fade away and disappear as the crystal is hit with X-rays. Crystallographers think of this loss of order and resolution as the death of their protein crystal and talk about crystals dying in the beam. With crystals of a smaller protein, you could collect all your data from one crystal, or if they started to die out with radiation damage, you could keep switching to new crystals until you had collected all your data. With ribosome crystals, you couldn't even collect your first diffraction image. Not only that, you had no way of knowing if the crystal had good enough resolution to begin with, because even before the first snapshot was completed, the spots might already have died out from radiation damage. At some point, scientists realized that cooling crystals would slow down the diffusion of free radicals generated by X-rays and thus slow down the damage. The first real evidence that this worked was produced when David Haas, who had been a postdoc at the Weizmann Institute in Israel, joined Michael Rossmann in Purdue. Like many of his generation, Rossmann also got his start at the LMB, where he worked with Max Perutz on the first hemoglobin structures. From there he went to Purdue, where he has spent the rest of his life and is now a world-famous scientist. Even now in his 80s, he maintains an amazingly active group and is so energetic that he is known to climb up mountains faster than many young postdocs and students. Haas and Rossmann decided to cool crystals of an enzyme to -75 degrees Celsius and noticed that the X-ray diffraction spots from protein crystals would die out from radiation damage much more slowly. But simply cooling crystals to low temperatures would not work in most cases. Crystals are about half water, which is why even though they can often look very regular with well-defined faces and edges, they are squishy like jelly or crumbly like cheese if you poke them with a needle. Remember, proteins are irregular molecules, and when they stack up, they touch each other at only a few points, and there are large channels of water in between. So when you take a crystal and cool it to very low temperatures, the water in the channels freezes and expands and ends up basically destroying the order in the crystals. Greg Petzko, who was then at MIT, found a way around this by replacing the aqueous solvent in the crystals with something like an antifreeze. In some cases, this would still preserve the order of the crystals. This allowed him to collect data at low temperatures and, as Rossmann had noticed earlier, with very little of the radiation damage that you would see at room temperature. For some reason, these methods didn't catch on until later, perhaps because they were quite difficult to generalize, so if people didn't absolutely need them, they just didn't bother. Meanwhile, electron microscopists, whose samples were also being damaged by irradiation with electrons, became interested in looking at samples at low temperatures too. Jacques Dubochet, who was working at the EMBL in Heidelberg, figured out that if he rapidly plunged his samples into liquid ethane, the water would cool so fast that there was no time for the water molecules to freeze by arranging themselves as ice. The water would vitrify or become like a glass, preserving the biological molecules in their native state. Meanwhile, a Norwegian from UC Davis named Håkon Hope had been working on collecting data on several small organic molecules that were sensitive to oxidation at room temperature. Perhaps influenced by Dubochet and others who were doing electron microscopy, he got the idea of coating his crystals with oil and then plunging them rapidly into liquid propane. The crystals maintained their order as he had hoped. Although we now routinely refer to the process as freezing and the crystals as frozen, it is important to remember that the method works precisely because the water doesn't freeze, but vitrifies. Håkon visited the Weizmann Institute in Israel, where he met Joel Sussman, who asked him if he could try to adapt his method for biological molecules. Håkon went back and used this approach on 2 very small proteins. He then returned to the Weizmann Institute and worked with Joel and his associate, Felix Frohlau, on making the method more general. One of the first crystals they tried was a crystal of DNA that Joel's student, Limor Joshua Tor, was working on. After that initial success, they got it to work on several other projects. Although by then Ada had a large operation in Germany, she continued to run a lab at the Weizmann Institute. At some point, Håkon and others in Joel's lab encouraged Ada to try his method of cooling crystals to help her with the problems of collecting data from her ribosome crystals. She was initially skeptical, but decided to give it a shot. It was not quite so straightforward to do it with ribosome crystals because they needed intense X-rays from a synchrotron, and cooling equipment was not standard on synchrotron beamlines at that time. Eventually, Ada and Håkon got some time to collect data at the Stanford Synchrotron Radiation Light Source, which is only a couple of hours away from Håkon's lab in Davis. Håkon put his cooling equipment in his car and drove down to Stanford and set it up. According to him, the first freezing experiment worked very well, and they could see a nice diffraction pattern with lots of spots. The next dozen or so tries failed, but that first successful attempt gave them the confidence that it could work, so they kept going until eventually the technique worked more reliably. Once the method worked for ribosome crystals, Ada became an evangelist for the cooling technique, which became known as cryocrystallography. Despite their success, the method took some time to be widely adopted, because their original method involved painstakingly sandwiching a crystal that is typically less than 1/10 of a millimeter long between 2 tiny shards of quartz that were glued onto the end of a pin. A few years later, in 1990, Qiuyi Teng at Cornell invented a simple procedure that involved fishing out a crystal from its drop with a tiny flexible loop at the end of a pin, where it was held in the loop by surface tension. This turned out to be much simpler even than the old method of mounting crystals in narrow capillary tubes, and led to a nearly universal adoption of cryocrystallography. It is often the case in science that just showing something works better is not good enough. It also has to be easy to use. The original crystals of the 50S subunit from H. Marasmortui were noticeably better than any that had been obtained, and after systematically tweaking the conditions to improve the quality of the crystals and cooling the crystals, Ada and her colleagues could now see diffraction spots out to 3 angstrom. This was well above the threshold needed to build an atomic structure. When the results were reported in the Journal of Molecular Biology, I had just begun my sabbatical and instantly recognized it as a major landmark. It was exciting to imagine that an atomic structure of the large subunit was now possible. Set against this was the fact that around 1991, crystals of ribosomal subunits, and even the entire ribosome, had already existed for several years, and even if they didn't diffract to such high resolution, they should still have been able to yield maps that were detailed enough, at least, to see roughly where the individual proteins and RNA were. And yet, there were no such maps. Perhaps the ribosome was just too large for crystallography. Still, the paper by Ada's group in 1991 got me thinking during my sabbatical. I started wondering what she would do now that she had crystals that diffracted so well. I was soon about to see where things stood. Every few years, the ribosome community meets in different parts of the world to catch up on the field. The next of these meetings was going to be held in Berlin soon after my sabbatical. It was a sad occasion, because Wittmann, who had done so much to make Berlin a center for ribosome research, had died suddenly the previous year, and his colleague, Noord Nierhaus, had taken over organizing the conference. Although the protein S5 I had solved on my sabbatical was just a tiny piece of the ribosome, it was the first atomic structure of any part of the ribosome to emerge for several years. I'd never been to one of these meetings before, so Steve White generously agreed to let me speak about it. There was nothing I particularly remember from the meeting, and certainly there seemed to have been no major progress on the structure of whole ribosomes beyond what I'd already read. On my return to Brookhaven from my sabbatical, I was vaguely unsatisfied. After a year at the LMB, the contrast was striking. Oddly, in some ways Brookhaven was a lot like the LMB. Scientists worked in small groups and did experiments themselves instead of managing people, and there was steady funding from the Department of Energy, DOE. But the bureaucrats at DOE had little sympathy for the small-scale science that produced real breakthroughs in biology. If they were scientists at all, they generally came from a physics background and saw national labs as a place for large facilities like big particle accelerators or reactors. As a result, I could see that the biology department, which had produced some top-quality science, was being slowly squeezed for funds, which made it difficult to attract the fresh young blood needed to maintain the vitality of an institution. A few months after I returned, I wrote to Richard Henderson, who was then head of the Structural Studies Division at the LMB, saying I'd enjoyed my sabbatical so much that I wondered if they had a permanent job for a guy like me. He replied that they had all enjoyed having me there, and while they all thought it would be nice to have me as a colleague, they didn't have any space or positions open, but I should keep in touch. I took this as a polite rejection. Around this time, Wes Sundquist, with whom I'd shared a lab bench on my sabbatical in Cambridge, invited me to visit Salt Lake City, where he had just joined the faculty at the University of Utah. They had a mixture of young, enthusiastic faculty and some well-known older ones, all in a beautiful setting surrounded by mountains. So when they asked me after my visit whether I'd be interested in a faculty job there, I was pretty excited about the possibility. Suddenly, 2 other positions opened up at the same time, but given both the people and the setting, I felt Utah would be the right place for me. They made me an offer that was 50% more than I was making at Brookhaven, and it made me a little uneasy because I thought I couldn't possibly justify that kind of salary. Soon after I said yes, all my fears about funding kicked in. At Brookhaven, even if you didn't get any outside grants, they would pay your salary and give you enough money to do science with a technician or two. At a university, you'd be dependent on getting federal grants, and I had nightmares of losing my grants and having my career go downhill. So I called up Dana Carroll, the chair in Utah, who had been extremely warm and friendly, to say that I was sorry, I wasn't coming after all. He was not happy. Soon afterwards, the starkness of staying on at Brookhaven also sank in. Apart from the problem of DOE slowly strangling the department, neither Vera nor I ever really liked living on Long Island, having to drive around everywhere and dealing with hot, muggy summers and cold, damp winters, which made my asthma worse. Rather contritely, I called Dana again and asked, could I change my mind again? He graciously agreed, but pointedly said he wasn't going to jump up and down this time. One reason for my initial hesitancy was that a germ of an idea was slowly forming in my mind, but seemed so risky that I wanted to be secure enough in my personal situation to have the guts to tackle it. During my sabbatical, I had used the MAD method to solve the small GH5 protein, and had been surprised by how such a small anomalous scattering signal from a selenium atom could give such beautiful maps. Could you use MAD to solve something really huge, like a ribosome? It turned out that ribosomes don't have a lot of methionine residues, so the signal would have been too low. But Wayne Hendrickson, who had pioneered using selenomethionine, had also used a different kind of atom for one of his structures. When he used holmium atoms to solve a protein structure using this method, his maps were not just good, but spectacular. The reason is that holmium and other lanthanide elements have a much larger anomalous scattering at particular wavelengths. Could you use one of these lanthanides to solve the ribosome? When I did the calculation, it turned out that you would only need about a dozen of these atoms bound to a ribosomal subunit to get the kind of signal you'd get from a typical protein that had already been solved using MAD. I already knew that many of these lanthanide atoms bound to ribosomes in a dozen places because a recent paper showed exactly that. I could hardly contain myself. This might be the secret bullet that would give me a route to solving the structure of something as large as a ribosomal subunit or even the entire ribosome. I redid the calculation several times to make sure I wasn't fooling myself, but the answer was always the same. If I had decent crystals, just a dozen or so of these metal atoms bound to a ribosomal subunit should give me its structure. When I was mulling over this idea, I remembered my encounter with Ada's lieutenant, Francois Franceschi, a Venezuelan of Corsican descent, whom I met for the first time at the conference in Berlin. Francois was very friendly, and took Steve White and me to see his lab in Whitman's old department at the Max Planck Institute in Dahlem, a posh area of West Berlin with lots of famous scientific institutes that predate the war. It was the same institute where Steve had worked before coming to Brookhaven. During our chat, Francois told me that their work was reviewed by a committee every few years. The latest feedback was that they were spreading their efforts too thin and should focus on the 50S subunit, now that they had good crystals of it. As I remembered this conversation, I realized that this meant that nobody would be focusing on the small subunit or the whole ribosome, neither of which had yielded good crystals so far. I thought the whole ribosome was premature to tackle, but the small 30S subunit, which binds the mRNA and helps to read the genetic code, was only half the size of the 50S subunit and seemed like a better bet. Suddenly, I sensed an opportunity to enter the big leagues, but it would have to wait just a bit longer, because I was about to move to Utah after 12 years in Brookhaven. In the meantime, there was another ribosome meeting to attend. Chapter 8. The Race Begins. The year 1995 was a watershed for both the ribosome and me. I was planning to move to Utah that autumn, so I decided to stop off in Salt Lake City on my way to Victoria, the capital of British Columbia, where the next ribosome meeting was going to be held. Vera and I spent our stay looking at houses in Salt Lake City and settled on one in the foothills with a spectacular view of the valley. We then went backpacking for a few days in the Hoh Rainforest on the Olympic Peninsula and took a ferry from there to Victoria, which was just across a strait at the southern tip of Vancouver Island. Victoria is a charming and picturesque city that appears to relish its colonial past, evident in its British architecture and landscaping. Right in the middle of our meeting, a large parade celebrating the birthday of Queen Victoria marched by outside. At the meeting itself, there was one exciting development and a surprising disappointment. The exciting development was seeing three-dimensional images of the ribosome from electron microscopy. The method had long been used to look at ribosomes and deduce their overall shape. But recently, researchers had started applying a method called single-particle reconstruction to objects like ribosomes that had no symmetry. Previously, the method had only been used to study regular objects like viruses. Biological molecules have very low contrast. Their scattering for X-rays or electrons is very similar to that of the water in which they exist. So previous work in electron microscopy had involved coating the particles with a heavy atom, like uranium, and looking at it in an essentially dry state. At best, you saw its surface shape, possibly distorted by drying out, and the level of detail was quite low. Despite the low contrast without stain, if you could extract enough signal from the particles, you might be able to get at the internal structure by electron microscopy. Richard Henderson and Nigel Unwin had done this with their two-dimensional crystals. It wasn't clear whether you could extract enough signal from the individual asymmetric particles, but as a result of work by Jacques Dubochet, you could look at biological molecules at very low temperatures by plunging the samples rapidly into liquid ethane. Just as with X-rays, the damage in the electron microscope is slowed down at low temperatures. You could expose the samples to a higher dose of electrons and see individual biological molecules even without stain. One of the leaders in the field was Joachim Frank, a German scientist who had been working away in relative isolation at the Wadsworth Center in Albany for years, where his lab was in the basement of a large government building. A tall, courteous, and somewhat reserved person with a deep love of art and literature, he even likes to write fiction and poetry, he also seemed quite insecure. Perhaps because, like me, he had spent much of his career outside the fast track of the big important centers of science. He was developing methods to extract a meaningful signal from the noisy images produced by biological molecules. At one point around 1980, he was joined by the Dutch microscopist Marin van Heel, who, with his outgoing and brash manner, was a striking contrast to Joachim. Soon after Marin went to work with Joachim in Albany, it became clear the town was too small for the two of them because he did not stay very long. Regardless of any other factors, their markedly different personalities would not have been a recipe for a prolonged and friendly scientific collaboration in any case. Marin ended up being recruited to Wittmann's Institute in Berlin, but during his short stay in Albany, he and Joachim managed to write a key paper that showed how to extract information by taking the noisy two-dimensional projections of particles in a microscope image and classifying them into groups that corresponded to different views of the particles. From that breakthrough, they both went on separately to develop methods to obtain a three-dimensional structure. The images of the ribosome that Joachim showed at the meeting were the most detailed we had ever seen. For the first time, we could directly see the tRNAs nestled between the subunits and the mRNA snake around a cleft in the small subunit. They were still at far too low a resolution to deduce atomic structures or even visualize an arrangement of the various proteins and RNA components of the ribosome. They looked like a collection of blobs that made up an overall shape. So much so that those of us who did crystallography and were proud of being able to get atomic structures would scathingly refer to the method as blobology. By contrast, progress on the crystallography was disappointing. Many of us wondered what Ada had done with her very good crystals of the large subunit that she had produced 5 years earlier. She reported that she had finally obtained phases with heavy atoms for her crystals and had maps that were at 7 angstrom resolution. At this resolution, the grooves for the double helices of RNA should become visible. Individual proteins, especially those that we had already solved separately, also become recognizable, and their structural elements, like helices, can be seen. But Ada's three-dimensional maps seemed to have no recognizable features at all. The audience, which consisted mainly of ribosome biochemists and geneticists, had no idea what to make of it. These kinds of specialized meetings are useful, not only because we hear about the latest cutting-edge work, but they are where scientists raise their concerns and debate with one another. This sort of frank give and take, even if heated at times, is what helps science move forward. So when the time came for questions, I raised my hand and asked, I know of at least 2 cases of important structures at 7 angstrom resolution. The first is Bacteriorhodopsin, in which the helices of the proteins could be seen as tubes, and the second is the nucleosome, in which you could clearly see the grooves of the DNA double helix. Since we know the ribosome has both, why aren't you seeing them in your maps? Ada replied, well, if the ribosome were as simple as Bacteriorhodopsin, it would have been solved a long time ago. And moreover, do you even know what the first maps of the nucleosome look like? The features we should see at a given resolution should not depend on the size or complexity of the object, but having made my point, I didn't want to prolong the argument. After the session, I was standing around with Peter Moore, Harry Noller, and a few others. Harry looked very contemplative and asked what we thought the problem was. We agreed that something appeared to be seriously wrong. The proceedings of the meetings are usually written up in a book, and Ada's chapter had the grandiose title, A Milestone in Ribosome Crystallography. I thought it was closer to a millstone. At least one part of it turned out to be wrong, the way in which the molecules were arranged in the crystal lattice, a knowledge of which is a prerequisite to be able to calculate phases and the structure. We didn't know it at the time, but in hindsight, this error may have been one of the reasons Ada's maps in Victoria were uninformative. I went away from the meeting thinking that rather than just contributing to the effort, I had a chance to make the next real breakthrough. Without my realizing it, Peter and Harry had come to the same conclusion. None of us said anything to one another at the meeting, but we all went back and began working on different aspects of ribosome crystallography. It was like the opening scene from the movie It's a Mad, 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 Mad World, in which, just after a car crash, an old man tells a group of assembled strangers that he buried the loot from a robbery in a park. The people all pretend that they don't believe him, but very quickly rush off to get a head start on the others. Any remaining reservations about tackling the problem were banished a year later during a meeting of the International Union of Crystallography in Seattle. Unlike the ribosome meetings, which brought together everyone who studied the ribosome regardless of the technique they used, this meeting brought together people who used crystallography regardless of what they were studying. I was invited to give a talk based on the work I had done on obtaining phases using the MAD method. It was the first time I had been asked to speak at an international meeting on crystallography, and as a relative newcomer to the method, I was naturally pleased. But another important reason to go was that Ada was going to be there. There were widespread reports that she now had crystals of the entire ribosome that diffracted to 2 angstrom resolution, which was astonishing. If true, it meant that her crystals were now so good that it would be quite difficult for us to compete with her. I was a little worried about having to speak to an audience of real crystallographers. These people had actually developed the method. I just used it. Fortunately, my talk went off reasonably well. Ada was scheduled to speak during a Monday morning session called Macromolecular Assemblies, which included a number of exciting talks about large, challenging structures. It was chaired by Wim Hol, a well-known Dutch scientist who had moved to Seattle recently. Each speaker was allotted about 20 to 25 minutes, with about 5 minutes for questions. The main job of the chair was to make sure that the speakers kept to the time and that the questions weren't dominated by just 1 or 2 people. Ada began with a short history of her work and then described her new crystals of the entire ribosome. She went into great detail about how she characterized them and why she believed they were ribosomes. She mentioned that they diffracted to 2 angstrom resolution. The diffraction pattern she showed was stunning, with lots of spots going out all the way to the edge of the photograph. We were hanging onto the edge of our seats, waiting to find out what she had done with them. She spent most of her talk describing how she tried to further characterize them before hitting us with the anticlimax. They weren't ribosomes at all, but a contaminant, which turned out to be a protein called enolase. By this time she had used far more than her allotted time, but Wim Hol was unable to stop her. She went on to describe her progress with real ribosome crystals. As far as we could tell, there had been hardly any since the previous year in Victoria. Several of us in the audience could not understand why Ada spent so much time talking about a contaminant. By the time she finished, the meeting was running over half an hour late. This had an unfortunate consequence for Paul Sigler from Yale, the last speaker in the session. Paul was a giant, both physically and scientifically. He too had got his start at the LMB and had solved a large number of very important structures during his career, becoming one of the most prominent structural biologists of his generation. A brash and confident man, he was known to be plain-spoken with a very hot temper. He once broke the glass panel on a Xerox machine by thumping his hand down on it in anger. On another occasion, he had smashed a desk drawer, but he was widely respected and admired. Paul had deliberately been put at the end of the program to cap the session because his lab had just solved the structure of GROEL, a large protein complex that helps newly made proteins fold up properly after they have emerged from the ribosome. But the moment he got on the podium to speak, some of the stage crew came on and hustled him off, saying, "Sir, you have to stop and leave immediately. We have a Nobel laureate speaking here in just a few minutes." Paul was furious. Apparently, the conference had organized a lecture to be given by a different Nobel laureate at lunchtime. And that day, it was the very same Hans Diessenhofer who, a few years earlier, had lectured in the Cold Spring Harbor course I had taken just 2 days before he got his Nobel Prize. All of us liked Diessenhofer, but, Nobel or not, none of us thought that he was in any way more worthy to speak than Paul. The buzz at the meeting was that Tom Steitz, a well-known crystallographer and colleague of Peter Moore's at Yale, had started working with Peter on ribosome structure. Tom had gone to Lawrence University, in his home state of Wisconsin, before doing his PhD at Harvard with Bill Lipscomb, a famous chemist who was also one of the early American protein crystallographers. There he met his wife Joan, who was working in Jim Watson's lab on ribosomes. Joan, today one of the leading molecular biologists in the world, first tried to work with a well-known cell biologist at Harvard for her PhD. But he refused to take her, saying, "But you're a woman. What are you going to do when you get married and have kids?" She could barely contain herself and burst into tears as soon as she had left his office. Fortunately, Watson had no hesitation in taking her on, and she was in his lab at a particularly exciting time in the very earliest days of ribosome research. Tom and Joan went to the LMB for their postdoctoral work, a common theme for many characters in this book. There Tom worked with the leading crystallographer David Blow on chymotrypsin, a protease, or an enzyme that cuts other proteins. Joan had a ruder introduction to the LMB. Watson had written to Crick asking him to find a place for Joan, but on her arrival at the LMB, Crick told her there was no space for her, and she could spend her time doing library research. Luckily, Mark Brecher shared some of his space with her, and Joan went on to make the hugely important discovery of how ribosomes get started at the right position on mRNA. A lot of socializing among academics happened during dinners at high table of the various colleges, so called because it is a long dining table on a slightly raised platform where only fellows and their guests are allowed to dine. Mark thought it was high time that women were included in this activity. At the time, most Cambridge colleges would not accept women as fellows, the only exceptions being women's colleges, such as Girton or Newnham. To get around this restriction, Mark proposed Joan as a member of the room at Gonville and Caius College, where he was a fellow. It was the first time a woman had the privilege of dining there without being someone's guest. By the end of their postdoc, Tom already had an offer from Berkeley, but on the way back, he and Joan had interviews at Princeton and Yale, and both universities gave both of them job offers as they walked out the door. As Tom describes it, "When I got to Berkeley, I put the 4 letters on the desk of the chair of biochemistry when I went to visit him in his office. I asked if there was any possibility of a job for Joan at Berkeley. He looked at the letters and looked at me and said, 'She's a woman. Women do not run their own lab. They work in the lab of their husband.' We went to Yale." They have been a star couple at Yale ever since, each excelling in his or her field. Joan pioneered many areas of molecular biology, including discovering the molecules called spliceosomes that chop and splice RNA in higher organisms before it is read by the ribosome. She would often get honors before Tom, like being elected to the National Academy of Sciences, and it is still a source of mystery to some of us that she hasn't yet been awarded a Nobel Prize for her work. For a long time, many of us thought she was on track to get it before Tom. When I first encountered Tom at Yale, he had already become one of the leading crystallographers of his generation. He was a burly, imposing figure who worked out at the gym with his friend and colleague Don Engelman. They both sported chin-strap beards, which made Tom look a bit like an Amish. Initially, Tom struck me as arrogant and pompous, but this was just my own deep insecurity showing, because later I realized he simply liked to be very direct, a characteristic of Midwesterners that I am intimately familiar with since I've been married to one for decades. Over the years we became good friends, and even though we ended up being rivals of a sort, he was always complimentary and supportive of me. But his directness, especially when combined with his enviable success in determining the structure of one incredibly important protein after another, did not endear him to some. Although many of us would marvel at his superb judgment and track record, occasionally I would hear the snide comment, "Well, you'd have great judgment too if you were married to a top molecular biologist." Being married to Joan may have helped, but those of us who knew Tom also knew he had a clear vision of trying to understand the underlying mechanism of the central dogma, or how information in DNA is replicated, copied into RNA, and used to make proteins. He had tackled nearly every aspect of the problem, even though some of his projects took years of persistence. So it was hardly surprising that he would want to tackle the ribosome. Learning that Tom and Peter were going to team up to work on the ribosome initially caused me some serious anxiety, because I was sure, with their combined expertise in the ribosome and crystallography, they would make formidable competitors. I was relieved to find out that they were focusing entirely on the 50S subunit, taking Ada's improved crystals from H. maris mortui as a starting point. I ran into Tom at the meeting in Seattle and asked him if it were true that he was going to go head-to-head with Ada. He smiled and said, "Well, we hope to diverge at some point." I too had very briefly considered using Ada's 50S crystals to try my phasing strategy, but didn't want to go into competition with her, partly because I thought people would not take kindly to my jumping on her problem. Of course, nobody owns a problem in science. Once results are published, anyone is free to follow up on them and even compete with the original discoverers. That right of anyone to pursue anything worthwhile, regardless of who began it or who else is doing it, has been very beneficial for science. But in the early days of protein crystallography, when both X-ray equipment and computers were quite primitive, and it took such a long time to collect data and solve a structure, there was a tradition that if someone had produced crystals of something, they were usually left alone to solve the problem. After all, there were plenty of other protein structures to solve. But the ribosome was different, because it was so important, and so many years had gone by since crystals had been obtained without much apparent progress towards an actual structure. This was despite the fact that right from the outset, Ada had strong support not only from Heinz Gunther Wittmann, but also from people like John Kendrew, who saw similarities between her goal of the ribosome structure and his own work on the first protein structure. She had a purpose-built lab near a synchrotron with lots of support from the Max Planck Society, including the operation in Berlin to make ribosomes and produce crystals. The Weizmann Institute had given her an exceptional long-term permission to work in Germany while allowing her to keep her tenured job and lab in Israel. The broader community, too, was appreciative. Right from the first breakthrough, she was regularly invited to speak at major international meetings. However, neither she nor anyone in her lab had solved large, difficult structures, nor was she collaborating with an experienced crystallographic group. So to me, it was as if someone who had never climbed a single major peak had decided to lead the first expedition up Everest without taking along an experienced Sherpa. The same bold stubbornness that allowed her to take on what appeared to be a nearly impossible problem in Wittmann's department and persist for years to finally get good crystals of the 50S subunit was now proving to be a hindrance in getting to the next stage. It was not surprising that 15 years after the first crystals were reported, even those who admired her efforts were getting frustrated and impatient. Still, now that Tom and Peter had gone public, I wondered how they would be perceived by the community. At a dinner during the meeting, I asked the well-known British crystallographer Guy Dodson what he thought of Tom using the crystals Ada had originally discovered to compete with her. He replied, without the slightest hesitation, that it was about time others started working on it. In any case, even though I had a small group with limited resources, I felt I had a chance to be part of the action. At least I wouldn't be competing with anyone, I thought, and certainly not with my old mentor, Peter and Tom. They could duke it out with Ada in a race for the 50S subunit, while I quietly carved out my niche by working on the 30S subunit. I returned to Utah with a renewed determination to crack the problem.
We've only got 2 chapters in, but that's pretty good. Getting started in Utah. This chapter in particular, I'm interested in the Utah work. I'll change the camera a little bit here. I'm in particular, and I still need to make some phone calls out to Colorado to learn about Jennifer Garrus, Garrus. She was one of the, she had a grant to work in the laboratory of Sina Bavari in 2001, 2, or 3, around that time. And her mentor and boss, if you will, when she went to the University of Utah was Wes Sundquist, Sundquist. Upon her murder in 2018, Wes Sundquist was the person who wrote the longest part of her, at least memorial, online, and what she had been working on, which was something called TSG 101 and the ESCRT pathways. Venki he was effectively one of the big names at Utah. And I noticed that when he had been doing, over the last several years, presentations of people that he had worked with at Utah, usually one of the 25 individuals photographed was Jennifer Garrus. So, of course, just to be clear, no foul play is being accused here. I'm just, at the moment I saw that Venki had worked with Wes Sundquist there, and this technology she had been working on had been identified as some type of an HIV blocker, I wanted to learn more about Venki. I knew immediately then, okay, this is a 1 in a million coincidence. This is obviously important. So I'm interested about the next chapter. I don't know why it's a pause there, but sometimes you just need to reboot a computer. Who knows? So with that said, thank you for watching. We will resume this perhaps later, perhaps tomorrow. And please, guys, take care. Put a smile on someone's face, please. And we will be continuing our work soon. I'm going to be streaming tonight, 7 p.m. Eastern Standard Time. It's going to be hosted on Twitter Spaces, Twitter Space live stream with Rebecca Smith, the mother of Danielle, will be joining us, and looking forward to that and the sites that she has set up and what has been new in her life and her family's life since we last spoke. So that's all, everyone. Thank you. Take care. God bless.